previously studied in the hemoglobin, myoglobin pigment also. In presence of carbonic anhydrase, the carbon dioxide along with water, it will form HCO3 minus and H plus. And why it is necessary to form these types of cations and anions? Because in blood, along with O2, carbon dioxide also available. And O2 is a moderate ligand and carbon dioxide is a strong ligand. So the binding tendency of iron with carbon dioxide is very high comparing to the oxygen. But CO2, this is not good for the biological system, or this is not good for the respiration system. That's why in presence of carbonic anhydrase, it will get converted to less toxic ion, HCO3 minus and H plus ion. And again, these HCO3 ions, it will carried by the blood serum, then it will go towards the lungs. And at lungs, again further HCO3 minus ion get converted to carbon dioxide and water, and through lungs, and I can say through nose, the carbon dioxide will go to the outer side of the atmosphere. So particularly this equilibrium reaction, it may be forward reaction or it may be backward reaction. It will be carried out by the carbonic anhydrase enzyme. So if carbonic anhydrase enzyme is absent, then this process, it will be little bit slower, or it will be very slower in presence of carbonic anhydrase enzyme or in absence of carbonic anhydrase enzyme. The rate reaction ratio will be just like 1 is to 340. But for our body or biological system, this ions requirement is very high. So naturally, carbonic anhydrase enzyme, it will present inside our body. That's why it will help to convert this carbon dioxide and water to HCO3 minus and H plus ion. So you can make a readout here also. Carbonic anhydrase affects the CO2 transport in the blood. Okay, it will make a conversion of CO2 into the some cation and the anion. You can read out here also. It's saying that the postulate is saying that the forward and hydration reaction happen in the erythrocytes during CO2 uptake by the blood in tissue, whereas the backward reaction occur in when the CO2 is released in the lungs. Okay. So particularly the forward reaction, it will occur in the muscle tissue, near the muscle tissue. Okay. And in the lungs, again the HCO3 minus ion, the backward reaction occurs, it will get converted to CO2 and water. And from lungs, through nose, it will go outside the atmosphere. Okay, so the carbonic anhydrase enzyme multiplies the rate of equilibrium by a million times. That means I can say it is a good amount of turnover frequency. Turnover frequency means if you can use a particular catalyst or enzyme for several times for a single function. Okay, and its molecular mass is 30,000 and it contains 240, 260 amino acids. You need to remember all the things. And another thing is that we'll discuss about its structure. So the structure of the carbonic anhydrase is just like a tetrahedral structure, and these are the protein chains. And at the last end of the protein chain, there will be a histidine amino acid. There here also a histidine amino acid. Here also a histidine amino acid. So on a protein change, at a particular point, this histidine amino acid is present. At a particular point, this histidine amino acid is present. At a particular point, this histidine amino acid is present. So particularly from the histidine amino acid, particularly the imidazole group. This one is called the imidazole group. In an imidazole group, there are two nitrogen atoms present. One of the nitrogen, it will get coordinated with the metal atom. Okay, or simply I can say this nitrogen lone pair, it will be donated towards the zinc 2 plus ion. Here also the similar thing. Here also the similar thing. And the fourth side, on the fourth side, it will get coordinated by a water molecule. And the overall structure of this enzyme will be just like a tetrahedral structure. Okay. So the basic function of the carbonic anhydrate is to get converted to CO2 to bicarbonate ion. So you need to remember about its structure. Structure is very important. And we will discuss about another zinc enzyme after some few minutes. This is called carboxy peptidase. And out of this carbonic anhydrase and carboxypeptidase, zinc, this carbonic anhydrase is more acidic comparing to this carboxypeptidase. Then we'll discuss about its mechanism, how it will get make a conversion of CO2 into HCO3 minus ion. And this is the mechanism cycle. So based on the experimental data, we can say that the binding of zinc atom to water lowers the pK value, and this makes water much more likely to give up its hydrogen ion and become a hydroxide ion. That means here, before coordination with zinc 2 plus, the O minus, the OH2 in water molecule, this oxygen's negative charge, it will shared by the two hydrogen atoms. That's why the release of these two hydrogen atoms is very difficult initially when the water molecule is freely available. When this oxygen atom get coordinated towards the zinc 2 plus, that means oxygen's electron density now it is utilized to form this sigma bond or this coordinated bond. Now here in near the oxygen, now the electron density is less, so it doesn't tightly bind these two hydrogen atoms. That's why we can say that before coordination, water molecule has some amount of pH value, and after coordination, this pH value will decreases. Okay, that means here it can easily, now it can, after coordination the hydrogen atom, it can easily remove from the complex. Yes or no? So before coordination, the pH value of water molecule is little bit high, but after coordination, this pH value decreases. So particularly we can say after the water coordination, its pK value or pH value will decreases, and it can easily remove a proton from this complex. Here you can see in the first step the H plus ion will remove from the complex, and it will form OH minus ion. So now this OH minus ion, it will be act as a good nucleophile. It will be act as a good nucleophile. So our aim of removing the proton from the complex is that we need to make a potential nucleophile, or we need to make a strong nucleophile. Initially, water molecule is a neutral ligand. It can act as a nucleophile, no issue. But when it will get converted to OH minus, now it is a very powerful nucleophile. Okay. Then it will interact with the carbon dioxide, the initially reactant carbon dioxide molecule, or I can say this O minus ion. It will attack to the electrophilic center here, the carbon one, and this charge will get converted to oxygen center, and it will form a complex like that. And this complex make a rearrangement to form this one like this. Here the O minus ion abstract this H plus ion. Here we'll get a O minus charge. Okay, and again this O minus charge, it will get shifted towards this point, and here a negative charge will create. It can make an interaction with the zinc 2 plus ion, just like that. Or we can form a regenerative structure like that. Now, at the end step or at the last step, here we can see also, if someone will ask you, in this transition state, metal is a pentacoordinated or tetracoordinated or hexacoordinated. You can see here one coordination site, here another coordination site, here another coordination site, here another. Now in this transition state, metal is pentacoordinated, but before the rearrangement, it is a tetracoordinated complex. After rearrangement, it will get converted to a pentacoordinated complex. And at the final step, water molecule will attack to this metal center, and this whole HCO3 minus or bicarbonate ion, this one HCO3 minus, it will remove from the complex, and it will form again the initial one. Okay, that means you are utilizing just a CO2 molecule along with water. Here addition of water, and finally we are getting HCO3 ion along with H plus ion. Okay, so this is the cycle, cyclic mechanism of the carbonic anhydrase. Okay, this is very important question. Most of the examinations will ask about carbonic anhydrase. These are the theories. You can make a reading also, or in your material also. All these things are available. Next, we will discuss about the another metalloenzyme. It's called carboxypeptidase, and it is here also the metal center is also zinc 2 plus ion. So first, we will discuss about uh, where these types of enzymes are available. Particularly, these are called the pancreatic enzymes. In pancreas, these types of enzymes are available. So what is the role of these types of enzymes? The basic role of these types of enzyme is to break the terminal peptide bond. To break the terminal peptide bond. You can see here, this is a structure of amino acid, and these types of bonds are called peptide bond, CONH. That means through this peptide bond, one amino acid is get connected to another amino acid. Yes or no? So here this amino acid will start with NH. That means it is called N terminal site. And the last one is COO minus. This is called C terminal site. So the aim or what the role of the carboxypeptidase is it will break the last amino acid from the C terminal site. This one. That means it will break this peptide bond and it will get converted to these two types of ions. So the function of carboxypeptidase is to hydrolyze, or I can say the breaking of peptide chain from the C terminal site. From the C terminal site. Okay. So different types of enzymes are available. Some enzymes it can break inside the chain also, but here particularly the carboxypeptidase enzymes selectivity is that it can break the last amino acid from the C terminal site. Okay, it's forming these two types of ions here. We can check here also the structure. Okay, here also it seems like a tetrahedral structure, but and before in carbonic anhydrase, you can see carefully the structure. Also here three histidine amino acid residues are present and one water molecule. But in carboxypeptidase, these two are histidine amino acid ratio, and this one will be the glutamic acid ratio. This one will be the glutamic acid ratio, and this one will be the water molecule. The difference is that here a glutamic acid is present, residue is present. Okay, in case of carbonic anhydrase, they are also there, but they are N-histidine amino acid present. The difference is here. In this place, in carbonic anhydrase, N-histidine residue is present, but here glutamate residue is present. Okay, you can see here the mechanism also. This is the enzyme, and here initially H plus ion will be moved from the complex. It will make a better nucleophile, and from this nucleophile, it is attacking towards the peptide chain. Okay, and from the C terminal side towards the C terminal side, it attack to the CO carbonyl bond NHCO. This is the peptide chain. I can say this one will be the peptide chain NHCO. It will attacking to the carbonyl group. Okay, and after that attack, this the oxygen atom will get the negative charge, and when the charge will return, there will be breaking of CNH bond. There will be breaking of this amino acid. Now it is split into two ions. Gets split into two ions. So in carboxypeptidase, we no need to mention all the figures. but you need to understand how the mechanism follows. Okay. Next two important enzymes are related to iron. One is catalase, another is peroxidase. So peroxide is a hemiprotein. So when I'm saying peroxidase is a hemiprotein, or I am saying hemiprotein means the iron is surrounded by a porphyrin ring, just like hemoglobin. In hemoglobin, in the planar surface, there will be four pyral groups uh, that will attack to the iron center. It will make just like a cyclic ligand. So hemiprotein means there must be a presence of just like a metalloporphyrin ring on the surface of the plane. And the role of peroxide is to the breaking of hydrogen peroxide into water and oxygen. Okay. So along with catalase also peroxidase is also same role. The catalase and peroxidase also has the same role, but its structure is little bit different. You can see here the structure. This is the structure of catalase. This is the structure of peroxidase. Okay. Here is a histidine amino acid ratio on the fifth side, and here is tyrosine amino acid ratio. That means on the fifth coordinating side, there are two different types of amino acids are present, except all other things are similar. And this Fe, this is called the metalloporphyrin ring. That doesn't mean here only nitrogen atom is present. And this is representing the pyrrole ring. This nitrogen, single nitrogen, that representing the pyrrole ring. This one. Okay. So simply the nitrogen means doesn't the simple nitrogen, the structure will be literally approximately just like that. There is a pyrrole ring, and the nitrogen is coordinated to just like copper center. Okay. That means on a cyclic ligand, okay, or a porphyrin ring. The nitrogen is from a porphyrin ring. That means here also the iron is surrounded by a porphyrin ring, and nitrogen is a part of the pyrrole ring. Okay. For representation, we simply representing nitrogen, and iron. That means this whole part it will get converted to, or it is told as hemi unit. This one, FeNN. So we can say this is a hemiprotein. This is a hemiprotein. Okay. You can see they also catalyze the hydrogen peroxide oxidation of the substrate, and the catalase have the largest turnover frequency with one catalyzed molecule converting million times of H2O2 to water and oxygen. So the main function of the catalase and peroxidase is to convert the peroxide molecules or organic hydrogen peroxides to the water and oxygen every second. So out of this catalase and peroxidase, catalase has the largest turnover frequency. That means it can utilize several times to make this similar reaction. And on the active side of peroxidase and catalase, iron 3 plus ion is present. Or I can say on the hemi-active side, iron Fe3 plus high spin complex is present. Okay, and the whole process it will occurs believed to occurs in two steps. In the first step, P means you can remember or understood just like porphyrin ring. That means it is consist of four nitrogen atoms. In the first complex, it will in the first step it will react with hydrogen peroxide. It will get converted to FeIP, P plus and plus here. So what happens in the first step? So in the first step, we can say this step will be a mesomeric form of this one, FeVP. That means Fe3 here it will get converted to Fe4. Yes or no? That means there will be oxidation of electron from the Fe. And what happens? Another oxidation Another oxidation occurs in the P site. You can see here initially P is present, and at the end of the reaction there will be P plus. That means two electron oxidation occurs, one electron oxidation from the Fe atom and another electron oxidation occurs from the porphyrin ring. Or I can say this whole complex representation. Sometimes you can represent like that. Or I can say these two forms are mesomeric form to each other. And in the second step, again it will react with another hydrogen peroxide. It will get converted to Fe3P along with a oxygen. Okay, and the process details about the process mentioned here. You can see Fe3P represents enzymes hemi group and Fe4P plus represents the mesomeric form of FeOVP, indicating that Fe3 is not entirely oxidized to FeP. Because Fe3 it is not completely oxidized to FeP. Fe3 it is get oxidized to FeIP, and the porphyrin ring is oxidized to porphyrin plus or P plus by losing one electron. By losing one electron. Okay. And the second step, it is further reacts with hydrogen peroxide. It will again get converted to Fe3P plus along with a oxygen molecule. Okay, so the whole process you need to remember about its mechanism. Then, what are the main properties of catalysts? Catalysts are only generally found in erythrocytes, plants, most aerobic bacteria. Okay, so these are the things you need to remember. These are just data you can remember easily. Okay, so different types of peroxidase are present: chloroperoxidase, lactoperoxidase, cytochrome P450. Okay. You can see the mechanism. Fe3 plus in porphyrin ring. It's make a attachment initially with hydrogen peroxide HOOH, and one of the hydrogen atom it will make a coordination with the nitrogen atom of histidine residue, and in the next step it will get converted to Fe4O, like that. Okay, better you can see here. In the first step, we are adding hydrogen peroxide H2O2. It reacts with water molecule. It will get converted to Fe plus O. And in the next step, further we are adding electron. It will also get converted to Fe4O. So initial also Fe4, final also Fe4. What happens here actually? So here this R plus is representing the porphyrin ring, or I can say this is porphyrin plus. Okay. If you add first electron, it will make a reduction of this Rs plus group. It will get converted to R or porphyrin group, and it will get converted to like that. And in the next step, if you add further electron, then it will get converted to Fe3O, and it will form water molecule here. Okay, like that. So you can represent the mechanism by these four boxes also, or you can represent simply the mechanism by the two-step reaction also. Next, we will discuss about cytochromes. So the cytochromes particularly found in the plants also, animals also, and here the active metal center is also iron. Okay, So they contain a hemi-like prosthetic group. So hemi-like means you can understand similar. There is an iron atom. There will be four chiral rings. It will get connected with by a CH group. And it will form a cyclic atmosphere or cyclic link. And inside the cyclic ring, there will be a metal center. Okay. So these particular types of groups are called the hemi groups. So in cytochrome, uh, the material suggesting that there is a hemi like a prosthetic group. That means this one will be the hemi group. Okay. And we can say this hemi group also we can classify it into different types, hemi A, hemi B, or hemi C. When I'm saying hemi A, that means this R1 group particularly. This one should be this CS double bond CS2. When I'm saying hemi 2, this one will be protoporphyrin. That means R1 and R2, it should be similar. So mostly when I'm saying hemi A types groups, hemi A types groups, this will available in the cytograms. Which types of cytogram? Particularly cytogram A. When I'm saying hemi B group, that means these two residues R1 and R2, it will be similar, just like this function. Particularly hemi B, it will available in hemoglobin also, myoglobin also, cytochrome B also, and peroxidase also. Or when I'm saying hemi C, this R1 and R2 group, it will be different. That means there will be presence of sulfur atom, sulfur residue, in the protein chain. And these are particularly found in the cytochrome C. So there are different types of cytochromes available. We can say A also, B also, C also. Okay. So in different cytochromes, the surrounding groups are different. So the flow of electron, particularly cytochromes, through the cytochromes, the flow of electron occurs to the biological system. What is the basic function or role of cytochromes? Through the cytochromes, the flow of electron occurs, and their redox potential value are designed such that, like that, you can see here 0.03, 0.26, and 0.4. The electron will flow from cytochrome B, starting from cytochrome B to cytochrome C and cytochrome A. Then this electron it will be trapped or abstracted by oxygen molecule, neutral oxygen molecule, and it get converted to O2 2 minus. Okay, and the electron flow step will be like that. Cytochrome B, you can check here the redox potential value will be very low. From B to C, it has the value 0.26, and then A has value 0.4, highest value comparing to all the cytochromes. The electron cyclic flow will naturally be like that. Okay, so when there will be flow of electron occurs, particularly from one cytochrome to another cytochrome, so there will be definitely a change in metal atmosphere. So metal atmosphere means sometimes metal will be initially iron 2, thereafter metal can be converted to iron 3, further it can be converted to again iron 2, like that. So particularly we will discuss about the cytochrome. Now we will discuss about cytochrome P450. So P450 means when you are taking this particular cytochrome at 450 nanometer, there will be absorption band will found in the UV spectroscopy. Okay, so this band is called Soret band. Particularly, this band is called Soret band, and this Soret band is the identification of cytochrome 450 because these types of bands appear at 450 nanometer in the visible spectrum region. Okay, and the basic role of cytochrome P450 is addition of oxygen. Sometimes these types of enzymes are called as oxygenase also. This called oxygenase. That means it makes a addition of one oxygen atom into the substrate. Or sometimes we can told it just like a monooxygenase. We can told it just like a monooxygenase because it introduces one oxygen atom into the substrate. While dioxygenase enzyme, it can insert two oxygen atom into the substrate. But the basic role we can say of the cytochrome P450 is addition of one oxygen atom into the substrate. You can see here And there is R. This is not O actually. This is RH. That means you are taking the alkane. This, it make an addition with the oxygen molecule, hydrogen ion, electron. It is forming the alcohol. That means formation of, that means conversion of hydrocarbon or RH to ROH. So here there will be addition of one oxygen atom. Okay, or simply you can say you can make an oxidation reaction also. And in next step, we can see if you are taking the initial substrate aldehyde, we are adding the oxygen, it is forming the carboxylic acid. That means there will be addition of one another oxygen atom. And alkenes also it can get converted to epoxide. Okay, and benzene it is get converted to phenol in presence of cytochrome P450. And amine it get converted to amine oxides. And sulfide it can get converted to sulfoxides. Okay. So these types of oxidation reaction it can carry by the cytochrome P450. Okay. And then we'll discuss about the structure of cytochrome P450. Okay. So in cytochrome P450, the iron will be always in Fe3 state and it is low spin octahedral complex. And the sixth coordination site it will be occupied by the water molecule, and another one of the S atom of 16 is coordinated to iron 3 plus instead of histidine in the proximal position. Proximal position means we can say this is the generally the hemicomplex. This will be the hemicomplex. On fifth side, fifth side means below the plane in place of proximal position. Proximal position means just below the iron plane. At a certain distance, there will be presence of a amino acid. And this amino acid is not histidine. This is cysteine amino acid. Cysteine amino acid means there sulfur can act as a donor atom. And on the above this iron atom, there will be a water molecule. The structure of the cytochrome P450 is just like that. That means it will form an octahedral structure. On the plane, there are four nitrogen atoms are present as a donor atom, and below the plane, there will be 16 amino acid ratio, and above the plane, there will be a water molecule that is coordinating to the Fe3 plus low spin complex. Okay, here are here the structure is present. You can check here. Structure of cytochrome P450. Fe3 plus, and this is the porphyrin ring, four nitrogen atoms on the plane that is surrounding to the Fe3 plus ion. When Fe3 plus means the ionic size of Fe3 is very less, so it is feed into the ring. If it will be Fe2 plus, it will form a dome type of structure. That means iron position will be little above the plane. But when it is Fe3 plus low spin, it will fit inside the structure. And on the proximal side, on the fifth coordinate side, there will be a 16 amino acid ratio. And on the top of the plane, there will be a water molecule. Now I can say iron it is forming a octahedral structure. The circular ring is called protoporphyrin or iron protoporphyrin, or I can say metalloprotoporphyrin. And if the particularly the metal is iron, I can say iron protoporphyrin. And this is Hemi B. Okay. So why Hemi B? You can check here. Two carboxylic acid groups are present, CH2, CH2, and COH. Similarly, here the surrounding residue is also CH2, CH2, COH. So that's why this is called Hemi B protoporphyrin. Because Hemi B protoporphyrin, I already mentioned here. Okay, here also CH2, CH2 group is present. I can say this Hemi B. Here there will be little uh, missing here. CH, CH, there will be COOH. Then this will be Hemi B. Okay, you can check here. This is a porphyrin ring. On the proximal side, 16 amino acid is present. On the offside water molecule is present. And this is the example also of the cyclic mechanism. Fe3 plus ion, 16 amino acid, water molecule. Initially, the hydrophobic alkane group, it will attack to the metal center, okay, and it will attach to this metal center, and the water molecule will remove from the complex. It will form a complex like that. Then further, there will be addition of electron, and when there will be addition of electron, the Fe3 plus it will get converted to Fe2 plus. Okay, and in the next step, there will be O2 coordination of a O2 minus ion to the metal complex. It will form O minus. Further, that means we are adding O2 minus. Further, if you are adding another electron, this O2 minus it will get converted to O2 2 minus. So when there will be O2 2 minus, they can form a peroxide linkage with the metal complex like that. This oxygen can coordinate it to this metal center, and this oxygen can also coordinate it to this metal center. Fine. And in the next step, further we are adding two protons, and these two protons that can remove one of the oxygen, and it will form a water molecule, and the complex will be like that. And in the next step, if we are adding the further water molecule, then this complete group RH and double bond O, this part will remove as just like a alcohol, and the further the water molecule get coordinated to this metallic center. So in the last step, the oxygen atom it will get inserted into this RS bond, and it will form a alcohol. And this ROH group it will get separated from this metal center. And in place of this ROH group, there will be addition of water molecule. Okay, so we need to check this mechanism steps very carefully. So these are all about the iron enzymes. Then we'll discuss about the copper enzymes. So particularly the superoxide dismutase, a very important copper enzyme. Or in short form, we can mention it as just like SOD. Okay. So in previous step, we will already discussed hydrogen peroxide will get converted to water and oxygen. So this formation of hydrogen peroxide it is occurs in presence of superoxide dismutase enzyme. From oxygen molecule and the proton atom, it can it can form the superoxide dismutase enzyme. It can form oxygen molecule along with hydrogen peroxide. You can see, and this first step, it will be catalyzed by superoxide dismutase enzyme. Okay. So in the first step, it is forming the hydrogen peroxide. But hydrogen peroxide is potentially we can say very toxic. That is not good for our biological system, and that's why in the further step, catalase it may converted to hydrogen peroxide into water molecule and further oxygen. Okay, so superoxide dismutase is different types. It may be nickel SOD, it may be iron SOD, or it may be manganese SOD. That means along with copper, if nickel is present, I can say nickel SOD. Along with copper, if iron is present, I can say iron SOD, means superoxide dismutase. Or if manganese is also present, I can say manganese SOD. Okay, so which types of SOD groups are available in different types of animals? Okay, you can check here. Some of the few examples: Fe or MnSOD is found in prokaryotes or the mitochondria of eukaryotic cells, while copper zinc SOD is found in the cytoplasm of eukaryotic cells. Okay, so in our body, in different part, these types of different types of enzymes you can find out different types of SOD enzymes. So particularly, these are three types: copper zinc SOD, manganese SOD, and iron SOD. So particularly, this copper zinc superoxide dismutase is very important, and this is the structure of copper zinc superoxide dismutase. And this copper and zinc, two ions, are coupled to this imidazole group of histidine amino acid. So here the copper structure is copper 2 plus ion is pentacoordinated. You can see here one coordination side, 2, 3, 4, 5. That means here the structure is square pyramidal. But this will be not a regular square pyramidal. There will be some distortion in the structure. This will forming a distorted square pyramidal structure. And here zinc is forming a tetrahedral geometry because the coordinating side is 4. So this is the complete geometry of the copper zinc superoxide dismutase. That is the molar mass of the copper zinc dismutase. Okay, so out of these two things, copper metal atom and zinc metal atom, which one is more important? Particularly, copper is more important. Okay, zinc ion here only provides the stability to the enzyme, but copper 2 plus it has great activity. It particularly participate in the reaction. Okay, so if someone will ask some examination, they will ask which one is more important, particularly copper ion is more important because the zinc ion just simply serve as a structural stabilizer by holding the bridging imidazole. तो गोटे imidazole को ये other side रे कौन करछ bind करके रखुछ structural खाली गोटे stability provide करछ. But majority of the activity or mechanism activity this will operate by the copper 2 plus ion. You can see here this is the initial enzyme. If you are adding one oxygen molecule, what happens? The O2 it will get converted to O2 minus. That means this proton here there will be protonation of hydrogen atom in this place. Okay. That means if you will treat this particular enzyme in place of in a acidic medium, there will be availability of H plus. So in this nitrogen atom, it prefers to bind with H plus ion comparing to copper 2 plus because this bond is just like a distorted bond because copper 2 plus has D9 complex has a distorted structure due to ZTD. Distorted structure means this bond length is bigger. That means this bond has less strength. So the nitrogen in acidic medium it will prefer to bind with hydrogen and this bond will break. Nitrogen and copper bond. And the oxygen it will receive one electron, it will get converted to O2 minus and further in the next step this O2 minus will abstracted one H plus ion, it will get converted to HO2 minus. And further we are getting this enzyme again. And in the next step, this HO2 minus it will abstract or it can make a addition with another hydrogen atom from this one. It is a hydrogen atom regeneration level. Okay, it is get converted to H2O2 hydrogen peroxide. This is one of the most important enzyme of the copper zinc superoxide mutase. Or you can see here the structure. Next, we will discuss about iron sulfur proteins. So simply, we can say these are the non-hemi iron proteins. That means, just like previous case, porphyrin ring, iron center is present in iron sulfur protein. There will be no presence of such types of porphyrin rings. That's why these are called the non-hemi iron proteins. First important thing about iron sulfur protein is these are the non-hemi iron proteins. Okay, and these are the memory based things you need to remember. Its molecular mass. Okay, and it contain 1 2 4 8 Fe atoms. That means in few iron sulfur proteins one iron atom is also present. In some other cases it can contain 2 4 8. So according to this iron atom there will be different types of iron sulfur proteins. Okay, and in between these iron atoms there are some sulfur atoms are present also. Okay, and here the arrangement will be just like a approximately tetrahedral structure. Okay, you can see the structures also. Iron, it is coordinated to on fourth side by the different sulfur atoms. It will form a tetrahedral structure like that. Okay, we'll discuss, but first you need to remember these things. So first types of iron sulfur protein we can say rubidoxine. Okay, and here in rubidoxine iron sulfur protein there are presence of one iron atom. There will be presence of one iron atom you can see in the middle, and along with four sulfur atoms. Okay, and the structure is just like a tetrahedral structure. It is not a planar structure. You need to remember this one will be the tetrahedral structure. And the sulfur comes from this cysteine amino acid residue site. Here one cysteine amino acid will get coordinated to Fe atom through a sulfur atom. Here another cysteine amino acid ratio. Here another cysteine amino acid ratio. Here another cysteine amino acid ratio. So when the sulfur comes from a amino acid ratio, then we cannot say this is a inorganic sulfur. We can say simply amino acids are the organic compounds. We can say these are the organic sulfurs. Okay. When sulfur is present in between two metal atoms that doesn't come from any amino acid, we can say this will be inorganic sulfur. But here there will be no presence of inorganic sulfur, only presence of organic sulfurs. You can see here, and it is forming a distorted structure, tetrahedral structure. And in the middle, the iron oxidation state is +3. So the inorganic sulfur is sometimes called as labile sulfur. Here there is no presence of sulfur in between iron, and these all these sulfurs are coming from the cysteine amino acid ratio. So in rubidoxine, four sulfur atoms are coordinated to one Fe atom, and the structure will be like that. And what is the function of iron sulfur proteins? Definitely, these are the electron transfer agent. These are the electron transfer agent. And this ruby reaction is one electron transfer agent because here one Fe atom is present, and this Fe it can get reduced to Fe2 plus and it's further oxidized to Fe3 plus like that. Here one Fe atom is present, so it can be one electron donor or one electron transfer agent. And another sulfur protein is called ferridoxine. Okay, so pyridoxine are different types: 2Fe2S or 3Fe4S or 4Fe4S. So first, we will discuss about 2Fe2S type of pyridoxine. 2Fe2S, and the structure of 2Fe2S is like that. That means two iron atom, and in between these two iron, the iron atoms, in between these two iron atoms, these sulfur atoms are called inorganic sulfur. These two are the inorganic sulfurs. But these four, 1, 2, and 3, 4, these are the organic sulfurs. So these are called inorganic sulfurs, or we can say labile sulfurs. When a sulfur is coming from an amino acid, we cannot say a inorganic sulfur. It is an organic sulfur. Okay. So when these types of iron sulfur protein is present, what will be the spin value? Suppose in the oxidized form, here also Fe3 plus, here also Fe3 plus structure is present, then what will be its d electronic configuration? So Fe3 plus means d5, high spin, d5 high spin. Then the electronic configuration will be in every d orbital there will be a single electron. Here also Fe3 plus, in every d orbital there will be single d electron. So these five unpaired electrons spin value it will be cancelled by these other five d5 electrons orbit electrons spin value. So here the net spin value will be zero. Net spin value will be zero. But in the reduced form, in the oxidized form, the net spin value is zero. I can say this is a it is providing a diamagnetic atmosphere. When there will be a diamagnetic atmosphere, EPR electronic uh, resonance spectroscopy EPR is inactive. You can say this part will be oxidized form will be electronic paramagnetic resonance part, spectroscopy part it will be active. Which one? This one. There is some spin value. When the spin value will be zero, when it will forming the uh, diamagnetic complex, it will be appear inactive. But on adding one electron, one of the Fe3 plus it is get converted to Fe2, and it will remain just like Fe3. Now tell me here, D5 electrons, here D6 electrons. So these five are cancelled by these five, and here will be extra unpaired electron. So the net spin value will be half. So you can say the reduced form is not diamagnetic. It contains one unpaired electron, and that's why it is forming a paramagnetic atmosphere. If there will be an unpaired electron, remain charge will remain, or it's forming a paramagnetic complex. This complex will be EPR active. It will be EPR active. So by using Moss bar spectroscopy or by using electronic spin resonance spectroscopy, we can differentiate in between its oxidized form of this iron sulfur protein and the reduced form of this iron sulfur protein. Yes or no? And this sometimes this 2Fe2 sulfur protein we can call is a risk center protein. So when there will be a risk center protein, there will be little bit modification in its structure. That means if these two amino acids, it will be replaced by two histidine amino acid, and structure will be like that. I can say this is a risk protein. Okay. ଏଠି ଦେଖ କ'ଣ ଅଛି ଦୁଇଟା ହିଷ୍ଟଡିନ୍ ଆମିनो ଏସିଡ୍ ଅଛି ଗୋଟେ ଏ ଷ୍ଟ୍ରକ୍ଚରରେ 16 ଅଛି 16 ଥିବ 2Fe2S ଫେରିଡକ୍ସିନ୍ ଯଦି ଏମିତି ଥିବ 2Fe2S risk protein. Okay, so next we will discuss about 3Fe4S or Fe3S for pyridoxine. So here you can see the structure of pyridoxine. Fe sulfur, this is inorganic sulfur, this one is inorganic sulfur, there is so Fe3S4 means 4S means you can understood in the complex in the structure 4 inorganic sulfurs are present. 1, 2, 3, 4. 3Fe 1, 2, 3. And the structure of 3Fe4S reduction will be like that. Okay. The structure is simple. And a part about its ECR activity. As the ECR activity, my EPR activity discussion kariba. Kon paiba. Okay. So he's mentioning that in its oxidized form, it contains 3 high spin Fe3 plus. Oxidized form in oxidized form. This one is also Fe3, this one is also Fe3, this one is Fe3. But in its reduced form, one of the Fe3 plus it is get converted to Fe2 and two other will remain possesses 2 Fe3. So that means it is a one electron transferring agent. ଯଦି ଗୋଟେ ଆଇରନ୍ ରିଡ୍ୟୁସ୍ଡ ହେଉଛି ତାମାନେ କ'ଣ ହେଉଛି ଆମେ ତାକୁ କହିପାରିବା this is a one electron transferring agent. So in the reduced form it contains Fe2 Fe3 ions and all are the high spin complex, all are the high spin complexes. So here in oxidized form also and in its reduced form also, if you calculate the total spin of the three Fe atoms combinedly, you didn't get anywhere spin value 0. If you are not getting spin value 0, you are getting some spin value. That means these types of complexes you can identify it or trace by using ECR spectroscopy, electron spin resonance, or we can call it EPR, electron paramagnetic resonance spectroscopy. Okay. Screen visible hochi.
Yes, sir.
Then we'll discuss about 4Fe, 4S. That means 4 iron atoms and 4 inorganic sulfurs. You can see here, this is a cube type structure. So when even number of iron atoms are present, we need to check whether the spin value will be cancelled by each other or there will be some paramagnetic against atmosphere. So in the this is the oxidized form. You can see here, this is the oxidized form and this is the reduced form. So in oxidized form, out of these 4 iron atom, 2 atoms are Fe3 and 2 atoms are Fe2. So definitely here one Fe3 it will be cancelled by another and one Fe2 it will be cancelled by another. So the total spin value is 0. Yes or no? When the total spin value is 0, we can say this is EPR inactive. That means we cannot trace these types of complex by using EPR spectroscopy. There will be some few spin value. It may be half or it may be 3 by 2 or 5 by 2. We can say this is EPR active. So we can say the oxidized form of 4Fe4S pyridoxine is EPR active, electron paramagnetic resonance active. Sorry, inactive due to its spin value 0. But when you see here the reduced form, so in the reduced form, there will be addition of one electron. When there will be addition of one electron, this Fe3 it will get converted to Fe2. You can see in the reaction. This is the oxidized form, and 4Fe4S is a one electron transfer agent. Initially, it will accept say electron. It will get converted to one Fe3, three Fe2. Initially, 2 Fe3, 2 Fe2. But after addition of one electron, it will get converted to 1 Fe3, 3 Fe2. So in the initial form, the net spin is 0. It will be pure inactive. But in the reduced form, after addition of one electron, this Fe3 particularly, it will get converted to Fe2. Now tell me, in this side, we can say the spin value is 0. But here, one Fe3 is present, one Fe2 is present. We cannot say this amount of spin, it will be cancelled by this one. So the net spin value will be half. So we can predict that the reduced form is EPR active. So most of the question they will ask from the EPR spectroscopy whether this protein is EPR active or not, whether this protein in reduced form is EPR active or EPR inactive, like that. In the long question also, they will ask these types of things. What is pyridoxine protein and what different types of pyridoxine proteins are available, and explain whether these are the EPR active or not in the oxidized form and in the reduced form. So this is all about iron sulfur protein. Then we'll discuss about nitrogen fixation. So definitely in the atmosphere, maximum amount of gas, diatomic gas is available. This is called nitrogen. Okay. But for our biological process, nitrogen is not available to our biological system in N2 form. We need to break the nitrogen. That means there will be decomposition of N2 molecule is necessary. Then it can get converted to porphyrin ring, it can get converted to hemoglobin, it can get converted to amino acid. Okay. So most of the important elements of our life, in the first sentence, you need to read out. Most of the important elements of life is available in the form of amino acid, protein, and porphyrin rings. Porphyrin rings means we can say it's a part of the hemoglobin. So these types of molecules we can form when nitrogen is available. But in nature, nitrogen is available in the form of diatomic molecule N2. So from N2 to amino acid formation, or simply you can say from N2 to ammonia gas formation, we need to break this bond in between the N2 compound. So in between NN, How many bonds are present particularly? Three bonds are present. Okay, so if three bonds are present, that means it is a highest bond dissociation energy. We need highest amount of, we need a higher amount of nature energy to break it. So we can make this breaking by two process, Haber process. This is the industrial process, and this is the biological process by taking particularly few bacteria also. But in Haber process, even there will be lot of amount of energy at atmospheric pressure maintenance is necessary along with a iron molybdenum catalyst. So this is very expensive. So here the formation of ammonia gas from this is nitrogen N2. This is wrongly written H2. N2, 3H2. They will combine. They will form ammonia gas. So this process is forward step is feasible, but the formation of here the ammonia gas is very expensive. That's why biological methods we can prefer the biological methods. So when nitrogen is get converted to ammonia, it can pass through two transition steps. Or in the first step, this N2 N2 N is N triple bond N. The first step it will get converted to NH double bond NH, and in second step it is get converted to NH2 NH2, and in the third step it will get breakdown to NH3 NH3. Okay, so when nitrogen is get converted to NH3, it will pass through these types of transition states. So when I'm saying nitrogen fixation means our aim is to make a conversion of N2 to NH3. Here read also the nitrogen fixation is the process that can convert the atmospheric nitrogen into NH3. Okay, so this conversion we can make by two methods. Now we'll discuss about after a few minutes we'll discuss about the biological processes, and the second method is the industrial process. So in industrial process, you can use iron molybdenum catalyst along with for maintenance of 400 to 500 degrees Celsius temperature and this amount of pressure, and it is forming these types of ammonia molecule, two moles of ammonia molecule. Okay, so in biological process, particularly in blue-green algae, you can say these types of in presence of these types of bacteria, Agitobacteria, Clostridium, okay, Rhizobium, that are found in the roots of the beans also. Okay, they can get converted the nitrogen into ammonia. Okay, naturally. There are other three types of enzymes are also present that can make this conversion from N2 to 3H2. These three types of enzymes are called vanadium nitrogenase, iron nitrogenase, molybdenum nitrogenase. But out of all these, molybdenum nitrogenase is very most important nitrogenase for the conversion of diatomic nitrogen molecule to ammonia gas. And these are the structural aspects you need to remember. You can see here the structure of P cluster of nitrogen. That means when I'm saying nitrogenase, it is consists of two different proteins. In a nitrogenase, it consists of two different proteins. In one part is called iron prosthetic group. Another part is called molybdenum iron protein. Okay. So there is a iron protein part along with there another is a molybdenum iron protein part. Okay. So this is the first part. I can say this is the P cluster of nitrogen. And P cluster of nitrogen means it consists of only iron, cysteine amino acid, or some few inorganic sulfurs. And this one, this part, this is the another part. So two groups are present. First group is called iron prosthetic group. That means this one, these two, and second part is the molybdenum part. This one. Okay, is mentioning that the Fe protein, this molybdenum nitrogenase consists of two different proteins. One is iron and another is molybdenum iron protein. The Fe protein has a molecular mass, this one, and the Fe protein has two Fe4S4 units connected by two cysteine ligand bridges. That means he's saying that the Fe protein it contains two Fe4S4 group by the two bridging ligands. And here the bridging ligand is the sulfur that comes from the cysteine amino acid ratio. Okay, these two units are linked by sulfur-sulfur bond. And in one FeSO4 unit, there is a serine ligand attached to one of the iron atom. So here you can see here there will be extra addition of serine group or serine amino acid ratio to this Fe. But here these types of interaction is absent. And the other part of nitrogenase that consists of these two FeSO groups, along with there will be a molybdenum group that is coordinating this iron sulfur protein part, and that is coordinating this iron sulfur protein. Or we can say this is the active site. That means when nitrogen will come, that doesn't interact with this iron sulfur, it particularly interact with this molybdenum group. That's why in nitrogenase enzyme or molybdenum iron protein nitrogenase enzyme, we can say molybdenum is the active site, not the iron active site. Is the active site because the reactant first interact with the molybdenum part, and then by performing some few experiments, it will get converted to NH3 and some ADP, PI, and H2. Okay, and this process is not clearly mentioned anywhere. There are different aspects how this reaction performs after addition of molybdenum. There are different mechanism aspects, but it's not clearly proposed. This N2 it's get converted to this NH3. You can see adenine triphosphate. It will get converted to diphosphate, and there will be removal of phosphorus. It will get converted to PI. That means this is the inorganic phosphorus that is removed from this ATP group. Here this is triphosphate. We can say it will be a combination of diphosphate and another phosphate. Okay. So these are the reactant steps. Along with there will be addition of 4 proton, 8 electron in all the steps required. Then finally it is forming ammonia gas, ADP, inorganic phosphorus, and hydrogen sulfur and hydrogen gas. Okay, so this is the enzyme that is responsible for the reduction of N2 into NH3 or formation of NH3 from atmospheric dinitrogen. Okay, you see here in the first step, this is a probable mechanism. In the first step, there will be addition of 2 electron, 2 hydrogen. It is get converted to N double bond NH2. And further, there will be addition of 2 electron, 2 hydrogen. It get converted to like that. And further, there will be addition of 2 electron, 2 hydrogen. It will get converted to molybdenum, and there will be removal of 2 ammonia gas. So in throughout process, there will be addition of a total of 8 electrons and 8 hydrogen. 8 protons and 8 electrons. Okay, so this is a possible mechanism, and but this structure is most important. We need to mention the structure. You need to remember the small molybdenum iron protein nitrogen structure. Then we will discuss about vitamin B12. Okay. So in vitamin B12, there will be some similarities, just like hemiproteins. This is not exactly hemiproteins, but in the structural side is little bit similar to the hemiprotein. Or we can say, in if I discuss about the vitamin B structure, B12 structure, the structure will be little bit like that. That means here the cyclic ring, this one, it will looks like a porphyrin ring. It looks like a porphyrin ring, but this is not exactly porphyrin ring. There will be some modification to a porphyrin ring. That's why this is called a corin ring. You can make this pronunciation. This is called the corin ring. Here the cyclic ligand is called the corin ring. Corin ring is a modified porphyrin ring with one fewer C double bond CH bridge connecting two pyrrole rings than the porphyrin ring. So what is the difference in between corrin ring and porphyrin ring? We can see here in the structure. There will be absence of extended pi bond. Here we can see there will be absence of extended pi bond. Another difference is that we can see the 5 member rings are connected by such types of group. But here these two 5 members are directly connected to each other. That means here, comparing to porphyrin ring, this ring is little bit compressed. This ring is little bit compressed, or I can say this cavity size is little bit decreases. And these types of modified porphyrin ring is called the corrin ring. And in the cavity, here the cobalt atom is present. So in you can say in vitamin B12 enzyme or in corrin ring, which types of metal center can fit easily. Here we can say cobalt ion, it can fit inside the corrin ring. Okay. And here you see here there is a on the upper side of the plane there will be attaching side. This is named as R. So if you add R in place of the fifth coordinating side above the plane of the cobalt, this will be called as cyanocobalamin, or we can say it is vitamin B12. If on the top of the side there will be OH group, we can call it as a vitamin B12 alpha. Okay. If in this place of R, CH3 group is present, we can call it as a methyl vitamin B12. Okay. So this is the complete structure of vitamin B12. Okay. And on the below of the plane, it will connected with a, another amino acid, histidine amino acid, and its total atmosphere is just like an octahedral atmosphere. So what is the role of the vitamin B12? Particularly, it will act as a group transfer reagent. That means these types of complex, suppose you can consider these types of complex, this X group, it can get transferred to this carbon position, and this methyl group, it can get transferred to here. So we can say this vitamin B12 coenzyme, it can make 1-2 rearrangements, or in simple language, we can say in presence of this vitamin B12 coenzyme, there will be 1-2 rearrangement or group shifting occurs. That means this X group, it will get shifted to this part, and this methyl group will come to this part. So simply, when some short question will ask, they will ask 1-2 type rearrangement or group shifting coenzyme for Charivati, something like that, vitamin B12. Okay, and in the active site, which metal center is present? We can say cobalt 2 plus. And the cyclic ring is not called hemiprotein. Cyclic ring is called corrin ring. And these types of shifting reactions are mentioned here. Some two group shifting reactions. We can see here B12 coenzyme presents a two position. No, this OH group will shift towards 3. That means from with respect to 1 to rearrangement, you can say if this is one position, this is two position, there will be shifting of one hydrogen to here, and this OH group shifted to here. So we can say there will be group shifting transfer reactions mostly occur in presence of vitamin B12. Vitamin B12 coenzyme, and these are the application of vitamin B12. So first thing in all metal enzymes, we need to remember its function, its structure, its active site. Okay, these things are important, very much important. Then we will discuss about photosynthesis. So simply in photosynthesis, you know, you are forming the uh, carbohydrate or glucose. You are forming the glucose or carbohydrate from carbon dioxide and water. Okay, this is the final reaction of photosynthesis. We'll discuss it. Carbon dioxide, adenine, adenine triphosphate, NADPH, proton, water, it is forming glucose or carbohydrate. Okay, and along with some few energy. Okay. So the particular requirement requiring reactants are carbon dioxide and water. So particularly we can say photosynthesis is a redox process and it occurs in green plants also, cyanobacteria, algae that converts water and carbon dioxide into carbohydrate. And this process is, we can say also sometimes carbon fixation. We can say carbon fixation. And this carbohydrate released in the form of glucose or we can call it as starch also. So here there is a star mark. You can check. There is a star mark and it is forming a carbohydrate. That means this is not the formula of carbohydrate. This is the empirical formula. We can, if you can write the actual formula, this will be C6H12O6. C6H12O6, and CH2 is the simple representation of its empirical formula. So C6H12O6, we can call it as carbohydrate also. Okay, no issue. And CH2 doesn't mean this is the actual formula carbohydrate. This is the empirical one. Okay. So in this process, he's saying that there is the removal of oxygen molecule in photosynthesis process, and this oxygen particularly, this is comes from water molecule. This is doesn't comes from carbohydrate. It's a carbon dioxide. It comes from the water molecule. So star means we can make a isotopically labeled atom. That means particularly here we can take O16, here we can take O17 isotope or O18 isotope. We can perform the reaction, we can trace this oxygen atom going to either carbohydrate or it is going to either the oxygen molecule. Okay, so this is a redox process. That means one element or one compound must be oxidized, another compound must be reduced. So here the water molecule, we can say it get converted to 4H plus O2 4E. That means this reaction one involves the oxidation of water to oxygen molecule. Here oxidation occurs. Where oxidation particularly occurs? There will be release of 4 electron. When there will be release of electron, I can say this is oxidation process. But that means initially here the oxygen atom is in the form of O2 minus. O2 minus. When it is not converted to O2, that means here oxidation state I can say 0. That means O2 minus to O. That means there will be release of 2 electron per oxygen atom. So here 2 atom molecules are present. That's why we can say there will be release of 4 electron. And in the next step, carbon dioxide along with 2 hydrogen, 2 electron addition occurs. It will get converted to carbohydrate and half O2. In this step, the addition of electron occurs. We can say this is a reduction step. If you can combinely add the two step, we can say two carbon dioxide along with water, it is forming carbohydrate along with oxygen gas. Okay, so the process of photosynthesis, simply you can remember it occurs in presence of sunlight or energy. So for photosynthesis, sunlight or energy is necessary. That's why this process always needs a input energy. So we can say this total process is a endothermic process. Okay, this is a endothermic process, and throughout the process, it needs different types of pigments, or these pigments are called the antenna elements. Antenna elements means that can easily absorb this sunlight and that can continue the process of photosynthesis. So these antenna elements or pigments are chlorophylls, pyridoxine, cytochromes, manganese complexes, and copper containing plastocyanin. So all these pigments it particularly available in the antenna molecules, particularly available by the in the antenna by the antenna molecules. In the antenna molecules, so I can say it is available. Simply, I can say these are the available in the chlorophylls of the leaf of the plant. Okay, or these are available in the carotenoids also, in position of carotenoids also, and sometimes phycinogens also. Okay, so mostly the antenna molecules, these are, they make a cluster or pigment protein complexes, and they will form a photosystem. Photosystem means they can easily absorb the sunlight, and then they can make a reaction or transfer of electrons or a transfer of energy. But maybe they can continue, but initially they can absorb the photo energy or the energy from the sun. I can say this is available in just like H nu. H nu means they can absorb the energy in the form of photons from the sunlight. And this light energy, it can easily get converted to chemical energy in pigment protein complex. Pigment protein complex means this is a this is a combination of all types of compounds that I told a little bit before. Pyridoxines, chlorophyll, cytochromes, all these are present in the pigment protein complex. Okay. So photosynthesis particularly depends upon the presence of chlorophyll. Okay. So it particularly it occurs in chlorophylls that are available in the green plants or green leaves of the plants. And these chlorophylls, I can say it may be chlorophyll A or it may be chlorophyll B. And the chlorophyll structure will be like that. That is here I can say there will be cyclic ring. Yes or no? And you can also ask, sir, whether this is a porphyrin ring. Four nitrogen atoms are present, no issue. But along with four nitrogen atom, there will be structural difference. You can see here one double bond is reduced. Along with there is a addition of this five member ring. Along with this five member ring, on the side group there will be different types of group present. Yes or no? So this is not a porphyrin ring. Okay. So this is simply called the chlorine ring. Chlorine ring means we cannot say chlorine INE. This is called CHLOREN ring. This ring is called chlorine CHLORIN. The cyclic ring is named as chlorine ring due to some structural modification. And in the metal active side, magnesium is present. Mg2 plus is present. Okay. If the X group means this one. This is noted as X. ଆମେ X noted କରିବ ଯଦି X ଟା CH3 ଥିବ ତାହେଲେ ମୁଁ କ'ଣ କହିବି chlorophyll A. If X is CHO or aldehyde group, I can say this will be chlorophyll B. Okay. If some chlorophylls are also present in bacteria also, there will be some modification of structure also, that is in place of X if CH3 is present, and in place of Y COH3 group is present, and in place of Z if this bond is reduced, they can form also bacteriochlorophyll. And the structure will be bacteriochlorophyll. So these types of bacteriochlorophyll, it will available in some bacteria also, and they can do also photosynthesis. So different types of chlorophyll. Sometimes it will available in green algae also, and in bacteria also. Little bit different types of chlorophyll also available that can perform the photosynthesis reactions. So this is the general structure of chlorophyll, in which magnesium metal active site is present inside the ring. This is called chlorine ring, and magnesium is the metal active site. Okay, now get we'll start directly the reaction pathway. And this inside the chlorophyll, there will be different types of photosystems are available, and the reactions particularly we can divide it into two parts. One will be light reaction, another will be dark reaction. So initially the reactions that are available in the screen, P680, P700, these are P68. That means these pigments absorb energy, particularly or light energy from the sun, from particularly 680 nanometer. 680 nanometer. Here P700 means it particularly absorbs energy at 700 nanometer. That means all these reactions from here to here, these are the light reactions. After this step, and these reactions, it particularly occur in absence of light. These are called the dark reactions. And finally, this is forming the carbohydrate. And up to this part, I can say this is light reaction because this occurs, these steps are forming in presence of sunlight. So initially there will be oxygen evolving step. There will be oxygen evolving process where in presence of manganese metal, this one we can say, this one particularly the water molecule initially in the reacting complex, the water molecule it is get converted to O2 4H plus, and this manganese IV it will get reduced to manganese 3. That means it will make a donation of electron, and these electrons particularly donated towards the photos P68. That means photo system 680, and this photo system 680 or pigment 680 at nanometer, it will get converted to P star 680. That means this is the excited state and this is the ground state. And the electron that is coming from the oxygen evolving process system. This is called OES, oxygen evolving system. This one. That means from water molecule it can get decomposed to O2. That means O2 means here oxygen in a water molecule, oxygen oxidation state. I can say O2 minus. Here we are forming the neutral oxygen OO. That means these two electrons it will get shifted towards the manganese, and further manganese it is giving 4 electrons towards this P68, and it is further oxidized to manganese IV. That means 4 manganese. Here we can consider 4 Mn3 plus, it is get converted to 4 Mn IV plus, and these electrons are creating inside this oxygen electron evolving system. Then it is donated towards P68, and P68 after absorbing electron it will get converted to P680. That means here the excited state conversion, this is not due to the electron, it is occurs due to this absorption of energy, and these electrons just attached to the photosystem P680. Further, this will pass through this reaction paths, and this reaction paths are mentioned here also. You can see here initially H0 manganese 3 manganese IV is present. After addition of 1 H nu, there will be removal of 1 electron. We can get Mn3 Mn2 IV. Further, there will be addition of 1 H nu. There will be removal of 1 electron. It is get converted to Mn3 IV. Further, there will be addition of 1 another H nu. It is get converted to Mn3 IV. That means in every step there will be addition of photon and there will be addition removal of 1 electron. If you consider from S0 to complete step S4 and again come back to S0, in whole cyclic process there will be addition of 4 proton, there will be removal of 4 electron, and in the last step you can see there will be removal of oxygen molecule, okay, along with 4H plus ion. So in complete step from S0 to S5, here there will be removal of 4 electron, and the electrons carried out by the P680. This is carried out by the P680. So what happens when P680 absorbs energy? The P680 ground state or photosystem 2. This is called photosystem 2. This is converted to P star 680. That is the excited state. And further P star 680, it will get converted to P680 plus, and it removes 1 electron. And this 1 electron, it is absorbed by phenophytin, by the another pigment present in the cycle, phenophytin. We can see here in the next step, there will be availability of phenophytin. So after receiving 1 electron, phenophytin is get converted to phenophytin P minus. Yes or no? Then further, what happens? The electron is further transferred to plastoquinone and Q-plastoquinone, and this step will continue just like that. Plastoquinone and Q-plastoquinone means we can say hydroplastoquinone. Here we can see This is plastoquinone, this will hydroplastoquinone. That means the electron will carries or shifted from one pigment to another. Here you can see Q means quinone, hydroplastoquinone, S2Q. Okay, then thereafter there will be risk center. Before we will discuss in iron sulfur protein, there will be risk center. Then on this cycle, there will be presence of cytochrome B. Then there will be a presence of plastocyanin fragment. Then this electron will transfer to photosystem 1, P700 pigment. And here, again, it absorbs sunlight. It can get converted to P star 700 or excited state. Again, this excited state will release electron, and this electron will carried by the iron sulfur protein ferridoxin. And this ferridoxin, in presence of NADPH, it get converted to NADP plus. And further, the electron comes from this step. It will make a addition of carbon dioxide and water. It's get converted to carbohydrate. And this there is a complete process also here, detailed process here. And this process is called dark cycle. This complete process is called dark cycle. And up to this process, this is called light reaction. And these type reactions are called dark reactions. And here there is a little bit details about dark reactions. That means the electron when NADPH, it is get converted to NADP plus. Along with there will be breakage of 2H plus and 4 electron here. And these 4 electrons that is trapped by carbon dioxide and it is get converted to sugar or carbohydrate. So you need to remember stepwise these pigments, photopigments. Initially there is a OEC, oxygen evolving process, occurs here. Then the electron is attached to P68 system. It is fast get excited to P star, then it is converted to P plus 680, then it will remove its electron. These electrons are absorbed by phytophine, then it is transferred to cytochrome B, plastoquinone, hydroplastoquinone, then risk center, then cytochrome B, again and plastocyanin. Then it comes to photosystem 1. P700 means these pigments particularly they will absorb the energy from the sun, from the sunlight, particularly at 700 nanometer. Okay, and on this step you can see the iron sulfur proteins are present. Cytochrome B is the center pyridoxine. So the function of iron sulfur protein is to carry out the electrons. So this is all about photosynthesis. So you can make a reading of details note here also, and notes are also available in the e-Gynecosy material. Okay, so that's all for today's class. Thank you. Okay, if you have any doubt, you can ask. So we are ending the class.